Hi everyone. So I am Dr. Hariharan, Professor of Biotechnology. I welcome you all once again to my another lecture session. That is, today we are going to discuss about one of a type of vector called artificial chromosome. So in this lecture, I have divided basically this lecture into three major parts. One is the basics of about the vector. Second is the bacterial artificial chromosome, and third is the yeast artificial chromosome. So let us first understanding the basics about a vector. So first of all, the basic thing what we want to know is what is a vector. So vector is nothing but a carrier DNA molecule which helps to deliver the foreign DNA into a host cell. That is, a foreign DNA is attached with a vector, then it is delivered into the host cell so that it can be multiplied in large quantity and produce a desired protein. So that is the actually what is the function and what is all about a vector. So we can classify the vector into different types. So based on the source, we can classify this into four major types. One is the plasmid, which is an extra chromosomal DNA. Generally it is engineered and used. And second is the viral vector, otherwise we can also call as a bacteriophage vector. And third is the cosmid vector. So cosmid is nothing but is a combination of a plasmid and a phage vector to have both the characteristics. And the fourth one that what we are going to discuss elaborately today is the artificial chromosomes. So if any vector, whether it may be a plasmid, bacteriophage, cosmid or a artificial chromosome, it should carry certain key features. So what are the basic key features of a vector? If we see, that's all engineered vectors generally carry these three basic features. One, it carries an origin of replication, or it carries a replication part, initiate the replication process, so it can produce the clones in multiple copies. That is the first one it needs. And second, it needs various restriction sites where we can insert the foreign DNA. So generally the restriction site will be present, otherwise it will be modified in such a manner that can be attached. So these restriction sites generally attached from outside by a name called as polylinkers, otherwise we can also call it a multiple cloning sites. And third is, once I have inserted a foreign DNA, I have to identify the vector which is a wild one, which contains the inserted DNA molecule. So this difference can be identified with the help of a selectable marker. These are the three prominent features a vector should carry. That is an origin of replication, the restriction site, and third is a selectable marker. So based on this, all the vectors has been put into one or other manner. So let us study elaborately about the artificial chromosome. So everybody knows a chromosome is a large molecule of a DNA generally present. As in humans, we can see that we have 23 pairs of chromosomes. Each chromosome carries different genes for our purpose. So we can synthesize this artificial chromosome for the purpose of inserting a large size of DNA. So after the invention of different vectors, there was an idea that how much large size DNA can be packed. So for the insertion of large size DNA, three things have been developed subsequently as a vector. So the first one is the pack, second one is the BAC, and third is the YAC. And later on we are also having a vectors of mammalian chromosomes also. So these BACs, PACs and YACs have provided a choice to insert a very large segment of DNA, more than 1000 kilobase pair. That was a major advantage. So before entering into an artificial chromosome, first we understand one of the top, that is what is a PAC. So PAC is a P1 temperate bacteriophage, actually carrying a packing site called as PAC. So this is extensively initially used to study the genomic characteristics of the E. coli. So this was the starting point where the further insertion of large size DNA molecule was possible with the identification of the artificial chromosome. So there are different types of artificial chromosome. The very extensively what we have understand is the BACs and YACs. BAC stands for bacterial artificial chromosome and YAC stands for yeast artificial chromosomes. So these are the two major artificial chromosomes has been extensively started using the as a vector in the human genome projects. So let us study one by one. So first we will understand about BAC, which is called as a bacterial artificial chromosome. So BAC has been designed with an objective that it's like a plasmid. It is a single copy sex factor of F which can be inserted into an E. coli. So the objective of this BAC was used to develop for development for the mapping and genome analysis of very complex species. So that was the basic idea the BAC has been developed. So let us consider the characteristics of a bacterial artificial chromosome. So we will start one by one characteristic. So while we were discussing about the characteristics, what is present in the of a vector, that is, then it should carry an origin of replication. If you see that BACs carries a replication genes, especially the ORIS and the REP-E gene. So these are the two genes generally you can see from the figure, that is the ORIS and REP-E gene. These two genes generally needed for the unidirectional replication of this F factor, this fertility factor. So this fertility factor can transfer from one generation to another generation. So the ORIS and the REP-E gene are the two genes which is responsible for the replication of this artificial chromosome. So the next main character what we can see in the BAC is that the copy number. So the BAC should carry or maintain a particular copy number. So generally there are certain genetic sequence, especially the pair A and pair B, maintain the copy number generally around 1 to 2, because the higher copy number leads to instability of this chromosome and we are not able to get the DNA in enough quantity. So as you can see from the figure that the pair A and pair B which is present next to the REP-E gene, so it helps to maintain this copy number. So generally a copy number of 1 and 2 is more than enough. So the third characteristics is the marker region. So once I insert the foreign DNA into the cell, I have to identify the clone from the ordinary wild vector or a vector which it doesn't contains the foreign DNA. So since it is like a plasmid, the bacterial artificial chromosome, it contains a antibiotic resistant region called as CMR, that is the chloramphenicol-resistant marker. As you can see from the figure, the CMR region will be present on opposite to the pair B. So this chloramphenicol-resistant region is generally helpful to identify the cloned one from the non-cloned one by growing the E. coli after transferring the rDNA of this BAC artificial chromosome can grow in the minimal media. This minimal media contains the chloramphenicol. So the chloramphenicol region will destroy the chloramphenicol and favors the growth of this cloned one. So it acts as a marker. So next characteristics, if you see that it carries certain sites, that is lambda cos n and a p1 log LOX logs p. These two cleavage sites generally present for the lambda terminase and P1 CRE protein for the P1 LOX P. So these genes, why it is very important, as you can see from the figure, it is present next adjacent to par B, which has been extrapolated as cos n and logs P. These two genes are generally responsible for certain activities. That is, what happened to these is, the in-depth idea of this logs P site is that it leads to the circularization. Once the vector has been inserted into the E. coli, that is this bacterial artificial chromosome, once it is inserted into the E. coli, this forms a circular form. So the formation of this circularization of this packed DNA will be favored by the CRE enzyme, CRE. This Cree protein helps in the circularization with the help of the host recombinase enzyme, which is developed by the E. coli itself. So the circularization of this artificial chromosome in a stable form is needed, which is done by this especially LOXP gene with the help of the stimulation of the Cree protein. This is important for the stability of this bacterial artificial chromosome inside a E. coli. So next you can see from the major characteristics is the restriction site where we have to insert the foreign DNA. So we have certain cloning sites, as you can see from the figure also. We have two cloning sites, ones for HIN2 and BAMH1. Apart from this, there has been certain GC rich cloning sites also available, restriction sites, that is S-fill, Nortel and other excision sites, as you can see from the figure. So these excision sites is next to the chloramphenicol resistant region, easy for the insertion of the foreign DNA and the identification of the foreign DNA, identification of the cloned one. So apart from this, after we insert the foreign DNA, after doing the cloning, growing into a large amount, this foreign DNA should be identified while we are undergoing the hybridization experiments. So apart from the other sites, it has been included with T7 and sp6 promoter region, which generates an RNA probes which is necessary for the hybridization reaction during the identification of the cloned gene. So as you can see from the figure, there is two things, that is T7 promoter and S6 promoter, which is present next to LOXP gene continuously, will be help in the identification. So these two genes are present in between the restriction site, as you can see HIN3 and BAMH1. So this is the basic characteristics of a bacterial artificial chromosome. Once again I will summarize the thing is that the bacterial artificial chromosome contains an origin of replication site, especially two replication site, that is ORIS and the rep-E gene, 2 for the replication it has, and to maintain the copy number of 1 and 2 it carries other gene, that is the pair A and pair B gene, and for the identification of the clone it carries an chloramphenicol-resistant marker. And apart from that, during the identification of the hybridization, it carries RNA probe promoters, that is the T7 and sp6, and apart from this it carries the LOXP site, especially for the circularization of the back once it enters into the E. coli. Apart from this, it carries various restriction sites like BAMH1, HIN3, and the modern vectors also carries the ECOR1. So let us discuss about what is the various advantages the BAC is having. So the BAC can be easily transferred into an E. coli very effectively, and it can be maintained into the E. coli carrying the large genomic fragment of a animal or a plant, even a human one, up to 300 kilobase pair, with higher stability, high degree of stability for up to 100 generation. So this was the major advantage the BAC was having, so that the higher species genomic libraries can be effectively constructed within a short period of time. So let us consider few examples of a BAC vector. So the original BAC vector has been derived from PBAC108L. So from the PBAC108L, the BAC vector has been modified and it has been developed, and currently we are using one of the two major BAC vectors, that is P-below-BAC11, PBELOBSC11, P-below-BAC11 and PEC-BAC1. So these are the two vectors, BAC vectors, widely used for the construction of the genomic library. So what actually the plasmid has been modified, so it can be used as an BAC108L, is that the original cloning sites has been replaced with a LAC-Z gene. So the LAC-EZ gene carrying the multiple cloning sites. So with this insertion of this foreign DNA into the LAC-EZ gene will help in the easy identification of the cloned one with the help of the formation of blue and white colonies in X-gal medium, as we have studied while studying the plasmid vector. So you can see an example with this picture, that is P. bleobac2, which is carrying a base pair of about 7507 bases. So in this picture you can see it carries the origin of replication site, that is ORI2 and Rep-E protein. So these two genes helps in the replication of the artificial chromosome. Then SOP A, B and C will maintain it into the copy number of this chromosome. Then it carries the cos site. This cos site helps in the circularization of this vector inside once it enters into the E. coli. Then it carries two marker region, that is the LAC-EZ alpha and the CMR, that is chloramphenicol-resistant one. So you can have restriction site in these two things, so that which will be help in the identification of the cloned one. If I have restricted in LAC-EZ alpha gene, it will be help in the identification through a blue and white colonies. If I restricted the foreign gene into the chloramphenicol-resistant region, I can identify the cloned one once it is grown in a minimal media carrying the chloramphenicol. So based on this, there was various improvements of this BAC vector started working on from 1980s onwards. So the P below BAC1 was the first output of this one, followed by the other vectors. P below BAC1 has additionally two ECOR1 sites. These two ECOR1 sites were present, one in the LAC-EZ gene and one in the CMR gene, that is the chloramphenicol-resistant gene. So based upon your objective, we can clone in the respective location. Similarly, the PEC BAC1 has only one gene in ECOR1 site in LAC-EZ gene only. So later on, the BAC still started improvement by replacing the LAC-EZ gene with a SAC-B gene. So what was the objective of this LAC-EZ and SAC-B gene? Was the LAC-EZ gene is needed for X-gal medium for the identification of the blue and white colonies, whereas the SAC-B gene was especially permits the cells to grow in a sucrose containing media, which can be easily positively selected on this basis. So apart from this, recently the BAC vectors has given another modification called the retrofitting. So BACs has been constructed along with one of the insertion site of a transposon. So the objectives of this transposon is that to enable the genomic inserts to be modified after the cloning. Once we have done the insertion of the foreign DNA into the vector, then we have cloned inside a bacteria, still this transposon gene will help us to do some modification. This is known as retrofitting. So what is the benefit of the retrofitting we are getting? Is we can do the transfection, we can maintain the episome, and we can do some functional analysis. This is the major advancement what has been happened in the BAC chromosomes. So what was the limitation we can find with the BAC chromosome is that I can put it into the two major limitation is that one is that because the BAC is a very large plasmid, it's a very large extra chromosomal DNA, it becomes very hard to manipulate in vitro. So during in vitro manipulation, if some shearing occurs, it causes the instability of this vector, and moreover if we do a separation during a gel electrophoresis, it doesn't give a restricted motility, so the separation doesn't happen effectively. And moreover, since it's a very huge chromosome, so naturally it carries various restriction sites. So there will be a multiple sites, similar sites will be available, so the common restriction endonuclease can act. So that should be taken care while performing the BAC. So this is the basic understanding about the BAC. So BAC is one of the wonderful vector that has been developed, and it has helped a lot in the human genome project for effectively completing the project with a short duration. So the next advancement what we have seen in the artificial chromosome is the yeast artificial chromosome. So the BAC is a prokaryotic chromosome, artificial one. It is an eukaryotic development of the chromosome, that is the yeast artificial chromosome. So if you see the yeast artificial chromosome, it includes certain key features. So what are the key features of a vector should carry? Along with an eukaryotic one, it has additional key features. So I have only summarized in this one. What are the key features? It carries the site, that is the yeast telomere site. Second, it carries an centromere sequence, that is the sun. Then it carries an autonomously replicated sequence, that is an ARS. Then it carries a selectable marker, that is URA3, and it carries a multiple cloning site in which the various foreign DNA can be easily inserted. So you see in the figure, so I have put it in a linear form, this chromosome. In the picture you can see at both the terminal the telomere sequence will be present. In the center the centromere sequence will be present, and next to left hand side of the centromere for us its ARS will be present, that is the automatic replication sequence will be present. Apart from that, the identification marker URA3 and TRP1, 2 will be present. Then a multiple cloning site between the centromere and the marker region is present for the identification of the cloned one. So the major advantage of this YAC is that, so we can in the back we can insert a genomic sequence up to some 300 kilobase per effectively, but YACs has an ability to insert a foreign DNA up to 1000 kilobase pair. So our construction of the genomic library of a larger species containing higher sequence of DNA can be done very effectively with yeast artificial chromosome. But the limitation is that in practice only 50% of YACs have the structural stability. It is mainly becomes unstable once it is inserted into the yeast. Otherwise since it carries multiple cloning sites, so more than 1 or 2 foreign DNAs gets inserted, and sometimes it forms a chimeras fragment, then finally it causes a problem. So this is all about our artificial chromosomes. So I will thank you very much for your continuous support to my lectures. So this lecture you have understand about the basic about the artificial chromosome, especially BACs and YACs. Thank you very much. Bye.